So hello, everybody. Good afternoon, and thank you so much for this invitation. As you heard from my background, I've been at UCSF for a really long time. I absolutely love the institution, and I was delighted to hear that I'd be able to join your ranks as an emeritus faculty member when I moved into the NIH position. So I look forward to participating in this lecture series moving forward. And as Bill Wara mentioned, I'm going to talk about lupus today, and particularly some of the interesting work that's been done within our group and around the world to understand how genetics as well as epigenetic factors contribute to the disease. And we've been focusing on trying to understand how these factors relate to the really significant heterogeneity that we observe in these patients, and also provide clues to better understanding the pathogenesis. So just to summarize where I'm going to take you this afternoon, I do want to provide a little bit of background about the disease lupus and the heterogeneity, which is quite striking for those of you that aren't familiar. And then I'll summarize what we've learned over the past decade or so in the genetic contribution to the disease, including how it's helped us understand the relationship between genes and very specific disease manifestations. And then I want to shift and focus on an area of increasing activity and interest related to epigenetic factors in lupus. And in particular, I'm going to be describing work by us and others looking at DNA methylation profiles as an indicator of lupus risk and severity. And then lastly, I'll conclude by highlighting the fact that epigenetics may provide a molecular link to the environmental contribution to the disease, which remains really elusive and very difficult to study. So briefly, lupus is a systemic autoimmune disease. It's notable for striking heterogeneity. Virtually any organ system can be affected. And the picture shown here just illustrates some of the many manifestations. The skin is affected prominently and very different ways: photosensitivity, a facial rash, a scarring rash known as discoid lupus. The kidney is affected, particularly among those with severe disease manifestations. There are central nervous system manifestations, which can be quite devastating. Arthritis is common, has some interesting distinguishing features from rheumatoid arthritis. And one could go on for quite a while, in fact, a whole lecture talking about the really remarkable heterogeneity that we observe among these patients. So autoantibodies, very prominent in lupus. Perhaps more so than any other autoimmune disease. And what's been interesting, in addition just to the sheer number of these autoantibodies that have been identified, but many of them have quite specific associations with clinical or other manifestations. For example, some, the anticardiolipin antibodies, are associated with thrombotic complications. Anti-double-stranded DNA autoantibodies are associated with significant renal disease. And there are other specific clinical manifestations associated with these autoantibodies. And there's quite a lot of evidence that suggests that the genetic factors are more directly or proximally related to autoantibody production than necessarily to the syndrome that we recognize as lupus. So like most human illnesses, though, in addition to the kind of clinical heterogeneity, lupus is a complex disease in that it's very clear that many different genes predispose to the disease among individuals, but genetics doesn't entirely explain the disease. There's an important role for the environment, and there's a lot of variation in the extent to which genetic risk factors translate into disease among individuals. And I'll be highlighting some of these features, but the main point here is that this complexity really hampered progress in identifying the genetic contribution to the disease for many decades. And that very lengthy process of gene discovery is kind of summarized on this timeline, dating back to the 1970s at least, when the HLA variants were associated with lupus and other autoimmune diseases. And progress identifying other genetic risk factors was very slow. Complement genes were implicated, and then there were some candidate gene studies implicating some other genes, but it wasn't until, if you look at the very end of this timeline, around the year 2008 or so, when, as a result of new tools that were available for much more cost-efficient and comprehensive screening of the genome has it been possible to identify larger numbers of genetic risk factors. And I'll summarize that work here in just a moment. So this slide shows a picture of the first 4 GWA, that is genome-wide association studies in lupus. This represented a very, very important turning point in the discovery of genes for lupus and was matched by similar results and progress in other autoimmune diseases as well as other complex diseases. So there were 4 big studies published in the year 2008 that allowed us to understand much more about the genetic contribution to the disease than had been possible in the many decades preceding that. Now, I will note, as you'll hear, all of these initial studies were focused on patients of European ancestry, which for a disease like lupus was somewhat disappointing given the much greater burden of disease among almost all of the other populations, non-European ancestry populations. But work in those important populations has progressed as well, as I'll explain. So for those of you who haven't seen the way results in a so-called GWAS study are typically presented, this is referred to as a Manhattan plot because when successful, it resembles the Manhattan skyline. And what's shown here is the x-axis has the position along the entire genome. Each dot is a specific genetic marker. The y-axis shows the strength of genetic association. And what you're basically doing here is comparing the frequency of these, in this case, 300,000 or so genetic variants in cases versus controls, looking for regions of the genome where there's association evidence for linkage to the disease. And as has been typical for most GWAS studies of autoimmune disease, the really prominent peak there in the region of chromosome 6 is the HLA region. This graph is truncated, so if it was not truncated, that peak would go up off the edge of the screen here. And what was really exciting was the identification of some novel genetic risk variants in this single experiment, that previously would have taken decades to discover. And so fast forward a few years, and now we're looking at a meta-analysis of several GWAS studies for a total of almost 5,800 cases and over 13,000 controls. And just in the course of those few years and the additional sample sizes that became available, now we were looking at 63 distinct risk signals at 52 genomic loci. And you can see the Manhattan plot here, which has gotten much more interesting and complicated, still with a very strong HLA contribution. Once again, we're still looking at European ancestry disease. So what's also been revealed as this progress has transpired is, if you look at the genes that are implicated and what their function is and what pathway they function is, you can learn a lot about the underlying causes of the diseases. And so that has been really exciting. What the title of this study highlights is really strong support for both a role for the innate as well as the adaptive immune system. And that has been reinforced in all of the studies that have followed. And another remarkable result of these studies has been the extent to which genes identified, say, in a GWAS study of lupus, have also popped up in the course of a GWAS study of numerous other autoimmune diseases, even those that don't resemble lupus clinically at all. So just to clarify this slide, what is shown here, the red circle surrounds all of the genes that had previously been identified or linked to lupus in prior studies before the current study, which identified those novel gene variants within the green circle. But now in the orange down below are those genetic variants that at that time appeared to be relatively specific to lupus. All of the ones in the blue circle on top, though, were being discovered in the context of studies of other autoimmune diseases. So it was not expected that so many of the lupus genetic risk loci would be shared by a very diverse collection of autoimmune diseases. And that pattern has been reinforced over and over again. So this slide just highlights the extent to which some of these studies implicated specific pathways. This is the toll-like receptor type 1 interferon pathway shown very simplistically, but virtually every protein that's shown on this slide has been implicated in a genetic study. So irrefutable evidence of a specific pathway involvement. So this was very, very exciting for those who've been trying to understand the underlying causes of this disease with limited success prior to that. And just to take things one step further, this is a complicated figure, but once again, just to illustrate all that can be gleaned in a study such as this, what is shown here, and I'm not sure if you can see my mouse, I apologize if you can't, but I'll try to talk you through it. On the outer edges of this circle are the chromosomes, 1, 2, 3, 4, 5, 6, etc. And the specific genes that have been implicated in GWAS studies are indicated there, many genes. If you start moving inside with the inner rings, the next ring here with the colors varying from yellow to red to orange indicates the function of the specific genetic variant, whether it's an enzyme or a kinase or a transmembrane protein, to start providing a clue to the specific function of that genetic variant. If you take another step towards the inner part of the circle, what's indicated here based on the white, pink, and purple is whether it's a coding variant or an EQTL or something in between, something undefined at that point. And the most inner circle, the different shades of blue, represent how many other autoimmune diseases have also been implicated by that single genetic variant. So one study really sheds a tremendous amount of light in the underlying causes of the disease. So now, what about this remarkable heterogeneity though? So far, those studies were looking at groups of individuals classified as lupus versus healthy controls. Now we know there are many different variations on lupus in terms of how severe the disease, the prominent manifestations. So we were interested in trying to understand whether genetics explained some of that heterogeneity. And as more GWAS studies were performed in, importantly, cohorts of individuals that had very well characterized disease, we could start teasing this apart. So for example, in this study that was led by Sharon Chung, also at UCSF, what we did is we divided the GWAS data into two groups. We looked at the patients that were double-stranded DNA antibody positive in one analysis and compared that to the results among the lupus that were double-stranded DNA negative, that lacked that autoantibody. And as you can tell, just by a crude comparison of these two Manhattan plots, which, by the way, were similarly powered because the case numbers were almost exactly equal in these two groups, very different genetic architecture with much stronger genetic associations, particularly in the HLA region among the double-stranded DNA positive. So importantly, these are all patients who are characterized as the same disease, lupus, but clearly in terms of the underlying genetics, they're quite different. And that theme has been recurrent as we've looked in different ways at clinical and serologic subtypes of the disease. In this analysis, we're taking almost 600 patients with severe lupus nephritis and comparing them to about 1,400 lupus patients who do not have significant lupus involvement, renal involvement. So no controls in this analysis, just all comers of lupus. Let's compare the genetics of those that have significant renal disease to those who did not develop that disease manifestation. What's striking is that if you had been staring at these tables of gene names, you would quickly observe that most of the genes identified in this study had not been identified in the case control lupus versus non-lupus studies. In fact, a completely new set of genes are identified in this sort of an analysis. Note also how the HLA contribution, which was very striking and prominent in the case control analysis, is very modest here, which suggests that there's a certain set of genes that contribute to developing the disease in the outset, but that other disease drive whether renal involvement, for example, is a significant or prominent component of the disease manifestations that develop. And you can look at the disease in a number of different ways. This is a way of a molecular phenotype. You can divide patients according to whether or not they have high levels of evidence of interferon alpha activity. And you will identify another set of genes that explain that feature of the disease. So you can keep on drilling down into more and more specific subsets to identify more and more sets of genes that contribute to lupus in different ways. Now, I want to turn to non-European ancestry populations. As I mentioned briefly towards the beginning, they are the ones that share the majority of the disease burden and the severe, particularly severe burden around the world. So it's very important to be able to study the genetics in these populations as well. So I'll show you a few examples and what it's revealed. This is one of the first non-European ancestry GWAS studies. This is a meta-analysis of two Chinese East Asian cohorts of cases and controls. And most of the genes overlap with those that had already been identified, although the relative strength is quite different, with HLA being less prominent, this gene over here, TNF-SF4, being very important, TNF-IP3 also being more prominent. But all these genes in red had not yet been identified in European ancestry GWAS studies of lupus. So studying a different ancestral population provided an opportunity to identify a different set of risk genes that are more prominently important in that population. Similarly, another East Asian study, this time in Korea, 1,200 lupus cases compared to 4,200 controls, once again reveals some novel genetic risk variants that hadn't been on our radar screen up until that point. And once again, here, an even more striking difference in the relative contribution of HLA to other genetic regions compared to the European ancestry population. So by studying diverse populations around the world, we can get a more complete picture of genes and genetic pathways that contribute to the disease. Now, we've had an opportunity also to look at Hispanic, or that is individuals of Amerindian ancestry, admixed individuals in a GWAS study. An interesting picture emerges here once again. In addition to some novel genetic regions, such as this one here, IRF5 is the most strongly associated risk variant for lupus in this population, in contrast to the European ancestry patients, and the HLA contribution is quite modest relative to that. Now, one of the things we have since discovered, the question was, well, are these genes uniquely important to Asian or Hispanic populations, or is it simply easier to detect their contribution in those populations because of differences in allele frequency? What we've done since is to go back with these novel non-European ancestry genes and look in much larger cohorts of European ancestry populations. And the evidence is there, but we were not equally powered. We were underpowered to detect them in the European ancestry cohort. So emphasizing the fact that to fully understand the genetic contribution to the disease, we really need to be looking at multiple, large, and diverse populations of patients. So now to illustrate this point further, even if you look within the continent of Europe, there can be, and this is the best example that I'm aware of, the PTPN22 functional risk variant, quite striking differences in the frequency of the risk variant. Shown in this graph of Europe is the striking differences in allele frequency of this functional variant, which is quite uncommon in the south of Europe, very common in the north of Europe. And once again, depending upon where you're looking, you may or may not have identified this functional risk variant. And interestingly, these sorts of striking gradients are believed to reflect selection pressure. For example, in this case and others like it, perhaps the selective pressure from the plague or other infectious diseases that swept through Europe, and may have selected for variants that have have as a consequence an increased risk for autoimmune disease. So an interesting theory that has been the source of some population genetic studies. So now I want to shift gears and talk about what are we learning about epigenetics and the risk of the disease and disease heterogeneity. And I'll take a minute to explain what I mean by that. So epigenetic factors, or epigenetics, refers to chemical modifications of DNA that can influence gene expression and other important functions such as that. They're distinct from nucleotide-based changes, which are reflected in genotypes and which represent genetic risk. These are chemical modifications to DNA that influence levels of gene expression and can also be very important for that reason. Now, we've been studying DNA methylation, which is simply one form of epigenetic modification, in part because it has been the most accessible and easiest technology to apply in large numbers, large cohorts of cases. But there are other forms of epigenetic modification that may be equally or even more important. Now, methylation in this context refers to methylation specifically at a CpG site, that is a cytosine that's followed by a guanine. And typically, what happens in that case when this methyl group is added is that that decreases the ability for the gene to be expressed. So more methylation results in decreased gene expression. Now, it turns out there are many exceptions to this pattern, but that's the basic concept, that levels of DNA methylation are going to influence levels of gene expression and are interesting for that reason. And for the reason, as I'll illustrate later, the degree of DNA methylation can be influenced by factors in the environment, hence providing that link to the environment that's been elusive and difficult to study. So I want to show one example of how powerful, in terms of a phenotype, DNA methylation patterns can be. This has become a famous example. This fat, yellow, and obese mouse is an agouti mouse. It's the agouti mouse model, which has been used in many studies of diabetes and obesity. And normally, these mice have that phenotype, as I mentioned. However, if they are exposed to a very high methyl diet, the phenotype can transform in striking ways. The subsequent offspring lose the yellow coat, they are no longer obese, and they lose the diabetes phenotype. However, that phenotype, which can then be stable, can also revert back to the original phenotype with exposure, for example, to a demethylating agent. So I think this example nicely illustrates how important DNA methylation patterns or levels can be in a phenotype. And DNA methylation has been widely implicated in cancer and some other human diseases, and increasingly in human autoimmune disease. So we first got interested in DNA methylation and lupus based on this very small study published a full decade ago now, which looked at 5 monozygotic twins who were discordant for the disease. So one had lupus, the other did not, and a set of matched control individuals. And what these investigators did is they characterized these 15 people for methylation sites, methylation levels at 800 sites across the genome. That was kind of all that was technically possible at the time. And then they measured the levels of DNA methylation at each of these sites in the 3 groups: the twins with lupus, the twins without lupus, and the healthy control individuals. And they summarized the results here in this heat map, which I think you can readily see the striking difference in the degree of methylation in this set of about 50 sites, which were much less methylated in the twins that had developed lupus compared to their healthy twins and the control individuals. So the blue means less methylation, the red means more. And interestingly, the 50 or so sites that were significantly different across these groups coincided with or corresponded to genes which logically would be playing a role in an immune-mediated disease such as lupus. So this was really exciting. This is potentially a way to describe or to explain differences in disease status among genetically identical individuals. So this work has advanced. The next iteration used technology in which it was possible to characterize 28,000 methylation sites genome-wide, covering 14,000 genes. And once again, still a very small study, 12 cases, 12 controls. But you can see in the set of sites that were significantly different in the cases versus controls, some being undermethylated, some being overmethylated, a set of genes are identified that are thus implicated in the disease risk. Now, we had been interested in looking at not only using the current technology, which looks at hundreds of thousands, and now we can look at 800,000 of these sites genome-wide, but also looking in larger cohorts that had well-characterized disease and asking the question, okay, so DNA methylation patterns are clearly implicated in risk of disease, but what about the disease heterogeneity, the specific manifestations? So what I've shown here is a Manhattan plot, not for genetic variants, but for epigenetic sites. And we're looking at lupus patients that either do or don't have double-stranded DNA autoantibody production as part of their disease. And what's striking about these results is not only the large number that easily meet this statistical significance threshold in this study. Number two, the number of genes that are implicating the interferon pathway, those are the ones that are asterisked, but perhaps most interestingly, the number of novel genes that are identified in this sort of approach, genes that are implicated in this methylation or epigenetic association study that weren't implicated in genetic studies. And once again, if we look here at the HLA region, shown here as MHC, it's quite modest compared to these other genome regions. So it suggests that we will learn about other pathways if we look at epigenetics beyond what we had already identified in genetic studies. So I'll show you one other phenotype where, and this work was led by Sharon Chung, I've mentioned her here as well in a previous study. So here, what we do is we expand the methylation analysis and we compare lupus patients with nephritis to those without. You heard me talk about that in the context of a genetic study a few minutes back. Once again, in this case, it's 19 genomic regions that exhibit differences in methylation levels according to their renal status. And if you look at the identity of those gene regions, you get important clues to pathways that are explaining this disease manifestation. So now I want to talk a little bit more about DNA methylation patterns and how they might help us understand more about the environmental risk for disease. So normally, patterns of DNA methylation are stably maintained across cell division. And you saw in the case of the mouse model how the pattern persists across generations even. However, these patterns of DNA methylation can be disrupted. And one of the clearly implicated factors that can disrupt these patterns is oxidative stress. And this provides a direct link to different types of factors in the environment. And so increasingly, there's been this interest in looking at the environment as playing a role in disease through its influence on epigenetic dysregulation or epigenetic regulation. And I've shown on this slide the many environmental factors that had been implicated in lupus, typically from epidemiologic studies that lack the ability to demonstrate causation, but nonetheless really mounting evidence that implicates, in spite of the challenges here, specific factors in the environment that we think are playing a role. And so how are they influencing the disease risk? So what's become clear over the past 5 to 10 years as epigenetic mechanisms of gene control have been studied further is it's become possible to link very specific exposures, environmental factors, be it chemicals in the environment, drugs, UV light, viral and other pathogens, hormonal factors, stress, all of these environmental factors, which have been associated with lupus, can be directly linked to epigenetic methods of gene control. So that this concept and this evolving knowledge has really prompted additional work trying to identify the role of epigenetics and then work backwards to identify additional factors in the environment. And I wanted, so the way this is all coming together, this is all consistent with a very general framework for understanding a complex disease like lupus, in which you start out, in a sense, with inherited genetic predisposition. And then over time, presumably as a result of exposure to factors in the environment with epigenetic influences. Over time, evidence of autoantibody disease, subclinical disease develops, and then eventually, if there's enough strength in terms of genetic predisposition, and then depending upon the exposures and the epigenetic effects, you ultimately get disease that we recognize clinically. And so you can think about this simply as in three buckets. I've talked about the genetic contribution to the disease. We've made a lot of project there. We're increasingly understanding more about the epigenetic contribution to disease, particularly through the DNA methylation profiling. And factors in the environment have been difficult. How do you characterize the environmental risk? How do you measure it? At what point is it important? Prenatal, childhood, puberty, et cetera. Really difficult to study that. So we were excited to hear about a concept that some of you may have heard about called the exposome, which is meant to, theoretically, it represents a measure of all exposures of an individual in their lifetime and how that affects their health. And you can think about it as a collection of exposures that have some sort of resultant internal dose and a biological effect. Here's the metabolic phenotype. And I like to show this quote by Christopher Wilde, and I think this resonated strongly with us. There is a desperate need to develop methods with the same precision for an individual's environmental exposure as we have for the individual's genome. I would like to suggest that there is a need for an exposome to match the genome. And we were excited to learn that literally right across the hall from us at UCSF was a laboratory that Roy Girona was running in the Department of OBGYN that was developing methods that would allow us to characterize the exposome. But first, a little bit more background. Why are we interested in doing this? I've already told you about some epidemiologic support for environmental exposures in lupus. But of course, the other concern is that we have a tremendous increase in the amount of chemicals that are produced, and we lack a comprehensive process for evaluating all of the chemicals that get introduced into our environment, raising many, many concerns, as you're well aware, about the impact on health. So we were excited to have an opportunity to do a pilot study, thanks to funding from the Lupus Research Institute, in which we could essentially characterize the exposome in a well-characterized cohort of patients with lupus in the context of also characterizing their genetics and epigenetics, and try to understand, try to identify chemical exposures that were linked to DNA methylation marks and might be contributing to their disease status. And I will simply tell you a little bit about the cohort that was developed largely due to the hard work of my colleagues, Maria Delara and Janus Yazdani at UCSF, in addition to important contributions from Patty Katz and Christina Lanata. And this work was funded by the CDC and allowed us to bring in 350 lupus patients in the San Francisco Bay Area to very deeply phenotype them. They spent a day with us in each of their visits to collect samples that would allow for genome-wide DNA methylation profiling and genetic genotyping, and the collection of biospecimens to support future research. So we've utilized this cohort, and we are, in collaboration with Roy Girona's lab, applying an ultra-high resolution mass spectrometry approach that allows us to characterize, in a sense, the exposome in a non-targeted approach. So this allows for the detection of potentially any chemical that might be present in a biospecimen as a result of a more traditional targeted approach where you have to know what chemical you're looking for to measure it. So this was a very exciting opportunity for us. The method specifically, and this is still quite a mouthful, but here on this slide, liquid chromatography, quadruple time of flight mass spectrometry. So still difficult to say that, but that is the methodology that Roy is using in his lab, and we've been characterizing samples in this well-characterized lupus cohort, and very excited to see what that reveals about potentially some novel chemical exposures. So just to summarize, and I hope I'm doing okay on time here, just to summarize, over the past decade, there's been really tremendous progress defining the genetic contribution to the disease, lupus, as with other complex diseases. But we still have a long way to go before we've connected the genetic association results with the specific causal variant and defined the functional or biological mechanism. Nonetheless, this work has provided really important clues to etiologic pathways, allowed us to identify potentially promising new therapeutic targets. We understand a lot better about the ways in which genes influence specific disease manifestations that are likely to develop. And in the past few years, we've learned much more about how, in addition to genetic risk, epigenetic risk also plays a role in determining who develops the disease when and what the resultant phenotype might be. And lastly, this methodology that allows us to study the exposome in a much more rigorous and comprehensive way, I think will prove to be very rewarding as well. So I'd like to acknowledge just some of the many people that have contributed to this work. Some of these contributions weren't reflected in the slides I showed today, but in particular, some of my colleagues at UCSF, I mentioned Sharon, and some other colleagues, I mentioned Roy. All of these individuals have contributed in really important ways, in addition to our collaborators at other UCs, our industry partners, and our international collaborators in Sweden, Australia, and the International Lupus Genetics Consortium that did a lot of the GWAS studies, particularly in the non-European ancestry populations. And then we have benefited from different sources of funding. These projects are, in addition to being time and resource intensive, expensive. And so we've had to seek funding in many different areas to make this work happen. And I also want to, in closing, just acknowledge the commitment and contribution and cooperation and partnership with the patients. This is a family with 3 young women affected with autoimmune diseases, including lupus, and they just nicely represent the amazing partnership we've had with the patients in our studies. So on that note, I'm going to conclude here. I'll stop sharing my screen, and I'm happy to answer any questions. I'm not sure how you want to do this or whether there will be a moderator, but thank you for the opportunity, and happy to stay on to answer some questions you might have.